my boots on and lace them up. A hard work, not another day to work. A hard work, that's what they say. Hard work.
Well, great. Thank you for, hold on one second. Thank you for inviting me to speak today. I don't have quite the history lesson plan that Dr. Morris gave, but what I hope to do is provide you with some information about the current research that's going on in Dr. Manitou's lab. And I will be following up on some of the toxicology concepts that Dr. Morris talked about, just in a more kind of current context. So as Sharon mentioned, I'm a postdoctoral fellow in Dr. Manitou's lab in pharmaceutical sciences. So the work that I'm going to be presenting today is work that has been ongoing in his lab for many years. And it's regarding a drug called acetaminophen, which a lot of you are probably familiar with, and the cellular responses that are occurring in the liver during liver injury that's caused by this drug. So I want to start by talking a little bit about a more general topic, which is just drug-induced liver injury. So this is not necessarily specific to acetaminophen, but rather can develop following the use of many drugs, including both over-the-counter drugs and also prescription medications. So the toxicity that can occur here can occur through a variety of mechanisms, and a lot of them are largely unknown or poorly understood. And the reason that people are so interested in drug-induced liver injury is because it's one of the most common reasons, or if not the most common reason, why drugs or pharmaceutical agents are not approved by the FDA, or potentially why they get removed from the market. So I don't know how much you know about the drug development process, but before a drug gets approved by the FDA for use to go to the market and be used by consumers, it has to go through preclinical and clinical trials in order to be tested for toxic effects and such, like Dr. Morris talked about. So the preclinical trials are done in cell lines and animal studies, and then the clinical trials done in patients. And really, what they've seen is that most of the drugs that are removed or don't make it quite to market are the common reason why that happens is because they don't actually pass the tests in terms of liver toxicity. So many of them are damaging to the liver. On the other hand, there's also been drugs that the FDA has approved and then subsequently removed from the market as a result of toxicity or adverse side effects that they've seen after they've been taken by a larger population of people. So this is a really common reason why, or drug-induced liver injury is a really common reason why drugs don't quite make it to be a successful therapeutic. So more than 50% of acute liver cases in the US are due to some sort of drug-induced liver injury. And there's a couple different mechanisms by which drug-induced liver injury can occur. The first are the type A reactions that can occur. And these are reactions that occur in a very predictable fashion. So they are dose-dependent, meaning that as you increase the amount of this drug, the dose of the drug, the toxicity increases as well. And they're usually a known toxic effect based on the clinical trials or the preclinical trials that have been done on this drug. So normally these reactions occur due to excessive dosing. So the person has taken a supratherapeutic dose of this chemical, and they produce a very specific type of toxicity. On the other hand, there are type B reactions that can cause drug-induced liver injury, and these are the unpredictable reactions. So they typically occur at what was considered to be a therapeutic dose. So they're not typically seen in those preclinical and clinical trials that are done, and they're often missed because they are have a very low frequency. So they're not often seen until you introduce this drug to a larger population of people. And it's mostly because the mechanism is very different. So the mechanism for the toxicity usually in this case involves some sort of allergic reaction to a drug that is unpredictable from person to person. And it's directed towards different components of the liver, and therefore the toxicity that it produces is not really uniform or predictable, as I've mentioned. So many drugs can induce liver injury, but what Dr. Manichew's lab focuses on is a drug called acetaminophen. And the reason that we focus on that is because acetaminophen is the leading cause of acute liver failure cases in the US and worldwide. So this graph, although a little bit dated, shows the percent of acute liver cases that are related to acetaminophen. And as you can see, over the last more than a decade, there's been a really high incidence of acute liver cases that can be attributed to acetaminophen. So this is a really important human health concern. And of that large percentage of acetaminophen-related acute liver failure cases, 48% of them are due to unintentional overdoses. So I'll talk a little bit more about this on the next slide. But the other 44% are due to suicide attempts, and there's about 8% that are due to unknown causes. So for those of you who aren't too familiar with acetaminophen, I'll start by talking a little bit about the drug itself. So it's abbreviated often APAP, so you'll see this nomenclature throughout the rest of the slides probably. It's a very widely, it's the most widely used analgesic, which is just a fancy word for a pain reliever, and antipyretic or fever reducer. It's considered to be a pretty mild analgesic, meaning it's commonly used to relieve headaches, minor aches and pains. It's often found as one of the active ingredients in a lot of cold and flu medications or cold and flu remedies, as you can see here. It is also commonly used to alleviate some of the pain associated with inflammation, although it is not an anti-inflammatory drug. So it doesn't actually treat inflammation, but rather just the pain associated with it. I hesitate to say this next bulleted point here after Dr. Morris's talk, because I guess I would say that it's a relatively low risk at therapeutic doses rather than use the word safe, because we just learned that no drugs are safe. But at therapeutic doses, meaning the dose that they put on the bottle when they say 2 capsules every 4 hours or whatever the case may be, if you stick to that therapeutic dose, this drug is relatively safe, low risk. However, as soon as you, even just a mild overdose of this drug can cause an acute toxification and lead to pretty severe liver injury or liver toxicity, and can perhaps even be fatal. So the interesting thing about acetaminophen in terms of the unintentional overdoses that occur, there was a really high incidence of the unintentional overdoses. And one of the reasons for this is because it's present in so many combination drugs. So it's the active ingredient among another active ingredient in a lot of these over-the-counter pharmaceuticals. So in other words, you may be taking 2 different medications, not realizing that they both contain the maximum recommended dose of acetaminophen. So now all of a sudden you've taken these 2 things in conjunction with each other and you've doubled the maximum recommended dose. So this is something that is on the labels of all of these products in terms of it's listed as one of the active ingredients. They're not hiding anything, but a lot of people, consumers, don't necessarily read the packaging of the over-counter medications that they're taking. So this has been a huge problem with acetaminophen and has really been a large contributor to all of the high incidence of unintentional overdoses. So one of the things that they've tried to do over the last couple of years to try and reduce the amount of unintentional overdoses is that the FDA has put together a panel of experts, an advisory panel, to give them some recommendations as to how they can reduce this number of unintentional overdoses, or what kinds of things they could change about how acetaminophen is used in order to prevent some of this. So ultimately what this panel came up with is that there really does need to be a more strict set of regulations for acetaminophen use, and there also needs to be better consumer education. So this panel that the FDA compiled of experts ultimately made several recommendations. The first thing they recommended was that several drugs, which are prescription drugs that contain acetaminophen in combination with other analgesics, such as Vicodin and Percocet, that those be removed from the US market entirely. The other recommendation that they made was that the maximum recommended or dose that is given in drugs like Tylenol, Excedrin, and other medications that contain acetaminophen, that those be reduced, like NyQuil and Theraflu, that the maximum allowable dose in those be reduced for the consumer. So again, these were just recommendations by this panel, but the FDA took them seriously. They decided ultimately not to eliminate things like Vicodin and Percocet from the market, but they did lower the maximum dose. So in 2009, at this time, they reduced the maximum dose from 1,000 milligrams to 650 milligrams per capsule, thinking that that would help reduce, the overall amount that the consumers were taking. So it turns out that this wasn't really strict enough, and they revisited this issue 2 years later in 2011 and put an even more strict set of regulations on the use of acetaminophen. So in 2011, they required that the maximum dose be reduced from 650 milligrams to 325 milligrams. So it got cut in half at this point. And at this time, they also required manufacturers to improve their labeling. So they now had to put a black box warning on any drug that contained acetaminophen, and said in a black box warning indicating that this drug could potentially cause very severe liver injury. In addition to that, they also launched several public education programs and such to help inform consumers about the risks associated with this drug and the potential for overdose and liver injury as well. So what we've focused our research on in terms of acetaminophen is trying to get at some of the molecular mechanisms and the cellular responses that are occurring because of this injury. And so, we know that acetaminophen is going to be around for a long time. It's not going to be removed from the market. It's a very powerful therapeutic agent and has great benefits, which outweigh a lot of the risks associated with it. So we want to try and, we're interested in trying to determine some of the factors that can be used to help prevent some of the toxicity associated with this drug. So this drug is most toxic to the liver, as I mentioned before. I'm not going to do any real hardcore liver anatomy with you, but I do want to just give you some sort of point of reference. For the organ that I'm going to be talking about. So the liver is located in the right upper quadrant of the abdominopelvic cavity, as you can see right here. So the main function of the liver is detoxification. It is taking harmful chemicals out of the blood, removing them or detoxifying them into non-harmful substrates that can be readily eliminated from the body and excreted. So if you're looking at this liver here, basically how it would function is that the blood would enter into the liver and the blood would circulate around the main cell type in the liver, which is called a hepatocyte. And the hepatocytes would essentially remove all of the harmful chemicals, or as much of the harmful chemicals as it could from the blood, detoxify them, and then put non-harmful metabolites back into the blood so that they can be excreted from the body. So it is considered a metabolic and an excretory organ. And because we're going to be talking specifically about acetaminophen, I wanted to just review quickly the metabolic pathway of acetaminophen in the liver. Now, don't think I'm going to talk about chemical structures, because I promise I won't. But I do want to just quickly go over the concept behind the metabolic pathway that occurs in the liver. So when acetaminophen enters the liver, it is going to be first detoxified by binding to what are called glucuronides and sulfates. So this is producing a non-harmful metabolite that can be readily excreted. So these two pathways are wonderful for detoxifying acetaminophen. On the other hand, if once these two pathways become saturated, there's another enzyme called cytochrome P450 2E1, which produces a harmful or reactive metabolite of acetaminophen. So under normal circumstances, your liver is capable of handling this harmful metabolite as well. It uses another chemical called glutathione, which is stored in the liver, in order to bind glutathione to this NAPQI, which is the reactive metabolite, and cause it to be non-reactive anymore and non-harmful so that it can be excreted. So under normal circumstances, when you take a regular therapeutic dose of acetaminophen, this is all well and good and your liver is able to detoxify it. However, at a super therapeutic dose, now all of a sudden you have the generation of a lot of this harmful metabolite, this NAPQI, and now you are getting to a point where you've generated so much of this that you're going to deplete the amount of glutathione that's stored in the liver. So as soon as the glutathione is depleted, well, now you have this reactive metabolite that can't be detoxified. And that can react with cellular macromolecules in order to, and would ultimately lead to cell death. So again, the main cell type in the liver that is going to be affected here are the hepatocytes. So to give you some idea of what we call cell death in the liver, I've included, how'd you do that, this? Okay. No. Oh. I've included a couple histology slides here. Now I know this probably looks relatively foreign, but basically what this is showing you is the difference in what liver tissue looks like in a mouse that has not been given acetaminophen, which is shown up top, and what a liver tissue would look like in a mouse that has been treated with acetaminophen. So basically what I'm showing you here is a section of liver tissue. And these cells have been chemically stained so that their cellular structures can be identified. So for instance, you can see the outline of each hepatocyte as it's stained kind of like a pinkish color. And then you can also see the dark purple spots. The dark purple spots are the nuclei of the hepatocytes. So in this normal looking tissue, you can also see some of the vessels are shown in this slide. So the vessels are bringing the blood into the liver and removing the blood from the liver. So this is what's known as the hepatic artery and the portal vein bringing blood into the liver. The blood is then going to circulate past the hepatocytes in these little channels called sinusoids, which you can kind of see some of the channels here in white. And as the blood passes the hepatocytes are going to remove the harmful chemicals from the blood that's circulating through the through the sinusoids. So the blood would continue down and drain into what's called the central vein, and then that would be removed from the liver. So the important thing here is that the blood is passing directly past the hepatocytes, and that the hepatocytes are the main cell type that are responsible for removing those harmful toxins from the blood and detoxifying them. So again, this is a normal looking liver tissue, and then hopefully you can appreciate the difference between these two. So what you're seeing here is a liver tissue from a mouse that's been treated with acetaminophen. And you can see again, this is the hepatic artery and portal vein where the blood is coming into the liver. This is the central vein where the blood is leaving the liver. And if you look here, you notice that there's a lot of damage to the tissue surrounding that. And the way that I know that there's damage is because now I can't make out any distinct cells. I also can't see any distinct nuclei like I could up here. So all of the cells in this area have essentially died. So when I referred to cell death as being a product of that NAPQI, that reactive metabolite, this is what I'm referring to. So this type of damage is called central lobular necrosis, and it's kind of the hallmark damage for acetaminophen toxicity in the liver. So I just wanted to show you this as to point out one of the ways in which we assess liver damage in our laboratory. Yeah? Oh, okay. Okay.
All, and you can back up the slides, but if you want, but I know that might be difficult. I saw a cytochrome P450E2E1 back there that was creating the NAPQI.
Yes.
What happens if you drink grapefruit juice? More of this, less of this, more of this over a different time course? I mean, I know that the whole grapefruit and drug interactions is a thing, and so I was like, oh God, and if you're like, I don't know, I don't study that part of the problem, that's fine.
Yeah, I actually don't know, but it looks like Dr. Morris does know, so I'm going to let him chime in here.
Those enzymes in grapefruit juice works in a different one.
Oh, okay.
Oh, okay, good.
Oh, yeah, I'm seeing there's other ones. I'm over here on Love It.
Oh, okay. No, that's okay. Good, thank you for asking. So my main point in showing you this was to, just give you an idea of how we assess liver damage in terms of the animal studies that we do. Obviously, this is not an approach that you can use in a clinical situation. A patient walks into a hospital and presents with acetaminophen toxicity. You can't take a liver biopsy immediately and do some histology and take a look at see how much damage has actually occurred. So in terms of clinical applications, what we have are other signs and symptoms, biomarkers that can be measured in order to determine what stage of acetaminophen toxicity that patient might be in. So for example, this table illustrates the 4 different stages of APAP toxicity. The first stage taking place in the first 24 hours of the insult, where you have symptoms like nausea and vomiting that are induced. The second stage takes place 24 to 72 hours, where you get some abdominal pain in that right upper quadrant where the liver is located. In addition to that, you get these 2 biomarkers, AST and ALT, which are enzymes that are used as markers for liver injury. And those would be elevated in the blood. So in a clinical situation, they can easily take a blood sample, not a liver biopsy. And then stage 3 would take place 72 to 96 hours later. You get some other symptoms of liver failure, sometimes symptoms of renal failure as well, and you get a peak or a much higher amount of those biomarkers present in the blood. And then stage 4 typically occurs more than 5 days after the original insult, and it either results in the resolution of the hepatotoxicity, meaning that the toxicity isn't continuing or progressing any further, and the liver is going to start repairing itself and recovering from that injury, or it is going to be progressing even further. And at that point, if the progression continues, it usually leads to multiple organ failure, which can often be fatal. So the good thing is that if you go into the clinic fast enough after receiving or experiencing some sort of acetaminophen overdose, there is an antidote that can be used to treat this. So the chemical compound here that's used in the clinics is N-acetylcysteine. And again, it's used for APAP overdose, and its main function is to maintain or replenish those glutathione stores in the liver. So if you recall, the glutathione was really responsible for detoxifying that NAPQI, that harmful metabolite. And therefore, you kind of prevent some of the further injury from occurring. So the unfortunate thing is that it's most effective when you take it within 8 to 10 hours after the overdose occurs, which isn't always possible or isn't always likely to happen. So ideally, you want to get this in the system before any liver injury occurs, because the important thing to point out here is that this is not a treatment for acetaminophen-induced liver injury. It is going to prevent further injury from occurring, but it's not going to treat any injury that has already occurred. So you might be asking yourself, well, how do we treat drug-induced liver injury then if that's not a treatment? And the answer is, well, there really isn't a treatment for it. So there's no specific treatment for acute or chronic drug-induced liver injury. They can administer things like steroids, which can control some of the systemic symptoms that are associated with the injury. But ultimately, those steroids wouldn't really have any effect on the actual injury itself, meaning it wouldn't improve the injury or improve any chances of survival. In addition, I just talked about N-acetylcysteine. It's currently the only specific antidote that's available for acetaminophen poisoning, but again, it's not a treatment. It's something that you can take to prevent further injury. So ultimately, patients who have drug-induced serious, severe liver failure end up usually requiring some sort of liver transplantation as a treatment option. But again, there really is no drug that we can take or wonderful magical elixir that is available for treating this type of injury. So our research is not necessarily aimed at treating drug-induced liver injury, but rather learning ways in which maybe it can be prevented. So we use acetaminophen as a model for trying to figure out how the cell responds to this toxicity and basically trying to determine ways in which the cells can either develop resistance against it or protection against this type of toxicity. So one of the phenomena that's been developed in our lab, when I say our lab, I mean Dr. Manitu's lab, is the fact that there is a development of resistance to acetaminophen hepatotoxicity, meaning that several doses of acetaminophen will allow you to develop some sort of protective mechanisms. Dr. Morris talked about protection occurring in species as we evolve, but protection occurs even just beyond evolution. And basically what we've shown in previous studies in the lab is that rodents that are given a very low, mildly hepatotoxic dose of acetaminophen, when they're then subsequently exposed to a higher dose, if they've received that pretreatment dose, then they're somewhat protected against the damage that a otherwise lethal dose would have caused. So they developed this protective mechanism against acetaminophen by receiving a mildly hepatotoxic dose of acetaminophen to begin with. So this is a pretty interesting phenomenon, one that we study regularly in the lab. And it's also relevant to human health because it's also been demonstrated in the clinic. So there's clinical data that's indicating that patients also acquire resistance to repeated supertherapeutic doses of acetaminophen. And this has been demonstrated in several different ways. So I'm going to start talking a little bit more about our actual studies that we do in animals and how this protection may be occurring. So one of the papers a while back in Dr. Manitou's lab, they showed this what we're calling autoprotection. So the idea that a pretreatment dose of acetaminophen can provide protection against a subsequent higher dose. So one of the things that they've measured in order to do this is the plasma ALT activity. So the ALT, if you recall from a couple slides ago, was one of those enzymes or biomarkers that's used and measured in the blood in order to indicate some amount of liver injury. So this is one of the techniques that we employ in the lab to give us some indication of how much liver injury has occurred in these animals. So I'll start by just describing the dosing regimen for this animal study. So these mice are injected with either 400 milligrams per kilogram body weight of acetaminophen or the vehicle that acetaminophen is dissolved in, so that serves as a control. And then 48 hours later, the mice are either exposed to a higher dose of acetaminophen, 600 or 700 mg per kg, or the vehicle, again, as a control group. And then 24 hours after that second dose, the blood is collected and the ALT is measured in the plasma. So if you look at this figure here, it's showing the plasma ALT activity on the y-axis, indicating as an indicator of liver injury. And on the bottom, they're showing this challenge dose of acetaminophen. So the black bars here are indicating the group of mice that were treated with only the vehicle as a pretreatment dose. So this dose here, given at time 0, was vehicle only, no acetaminophen. Whereas the gray bars are indicating the group of mice that were treated with that mildly hepatotoxic dose of acetaminophen at time 0. So they received an APAP pretreatment dose. So what you're seeing here on the x-axis is the second dose, the challenge dose. So it was either 0 APAP, meaning that it was the vehicle, it was 600 milligrams per kilogram, or it was 700 milligrams per kilogram. So basically what this figure is showing is that the animals that were given the vehicle pretreatment had a huge amount of liver toxicity when they received this 600 or 700 milligrams per kilogram of acetaminophen. However, the animals that were given that pretreatment dose of acetaminophen, as you can see by the gray bars, had significantly less liver damage or toxicity, as evidenced by lower ALT values than the mice that were just given the vehicle pretreatment. So these mice were protected against this toxicity. So they were given the same challenge dose as the other group of mice, but they had significantly less toxicity. So one of the things is that we don't know is why, how are they protecting themselves against this toxicity? What is this first initial dose of acetaminophen doing in terms of a cellular response that is providing them with some mechanisms for protection? So one of the interesting findings that came out of those original studies in the lab was that there were changes that were occurring to what are called hepatobiliary transporters. So the hepatobiliary transporters is a fancy way of saying that there are proteins present in the membranes of the hepatocyte that are responsible for transporting substances in and out of the cells. So as I talked about before, where the blood was running through those channels, the sinusoids in the liver, and the hepatocytes were pulling things out of the blood, this is how they're actually doing that. They're using these transporters to take things up into the cell and then also to efflux things back out of the cell. So this schematic here shows a picture, cartoon of a hepatocyte. So what you're seeing here is the hepatocyte has what's called a plasma membrane to it. And the plasma membrane forms a barrier, a selective barrier between the inside of the cell and the outside of the cell. In this case, the outside of the cell is going to be contacting the blood. So this is what's known as the basolateral membrane that goes all the way around. And this is contacting the blood, or the blood that's running through those sinusoids that we talked about. The other membrane in the hepatocyte is the canicular membrane, which is shown here. And this is contacting the bile ducts of the liver. So basically what I wanted to just demonstrate here is that there are what are called uptake transporters, which are bringing substances from the blood, God bless you, into the inside of the cell. And then also efflux transporters, which are pumping different substrates from the inside of the cell back out into the blood or perhaps into the bile of the liver. So what they found from these studies was that in the mice that were treated with acetaminophen, they had differential expression, meaning that the amount of these proteins, these transporters, had changed. It had been altered because of the acetaminophen toxicity. So the hypothesis of this work was that the drug-induced liver injury is basically going to change the expression of these transporters, and that would in turn limit the chemical burden that's placed on the hepatocytes so that they can more readily be repair and recover from this damage. So there was two great papers that came out from this work several years ago from Dr. Manitu's lab, which described all of the different changes that occurred in these hepatic transporters as a result of acetaminophen exposure. So I'm going to just summarize some of the data from those papers here. So what I'm showing you is just some schematics of a hepatocyte. This would be the blood vessel. So these are the uptake transporters in the hepatocyte. And what they found in this work is that several uptake transporters, the expression of them or the amount of them present in the membrane was reduced when they were exposed to acetaminophen. In addition to that, they found that there were many changes in efflux transporters as well. So the efflux transporters, some were present on the canicular membrane here, and some were present on the basolateral membrane contacting the blood. So the expression of the efflux transporters had increased during exposure to acetaminophen. So all of these could be serving as protective mechanisms, but we still didn't really know anything about how they're being protective. So what is it exactly that these transporters are doing that is protecting the cell against that subsequent dose of acetaminophen? So what probably the most notable finding from this research was a protein called MRP4, which is an efflux transporter on the basolateral membrane. So there was a dramatic increase in MRP4 when the cells were exposed to, when the animals were exposed to acetaminophen. So this is just some of the work. I don't know if you can even see that, can you, or no? Maybe if we turn the front light down. Great, thank you. That was nice. Okay, so this is just one figure from that work showing how the MRP4 expression had dramatically changed in response to acetaminophen. So what I'm showing you in this first group of pictures here is a technique called immunohistochemistry. And basically what it is another slice of liver section. And now this time the liver section has been exposed to an antibody that is specific for MRP4. So this antibody is then fluorescently tagged, and that's what you're seeing as the green fluorescence in the second figure here. So anywhere where you see green fluorescence is indicative of where that MRP4 protein is located. So as you can see, in a control mouse liver tissue, there's virtually no or no detectable amount of MRP4 present. But when you expose a mouse to acetaminophen, now all of a sudden in the membranes of those hepatocytes, you see a significant amount of MRP4 protein accumulating in that tissue. And this is also demonstrated down here using another technique called a Western blot that employs a lot of the same ideas, using an antibody to detect a very specific protein. So here, this is shown normal liver tissue, normal being not treated with acetaminophen, and then the acetaminophen treated liver tissue. And essentially, the density or the darkness of these bands is indicative of how much MRP4 is present in those samples. So obviously, there's a pretty dramatic difference or dramatic increase in MRP4 in the acetaminophen-treated mice. The other thing that they found in these studies was that there's a negative correlation between the amount of MRP4 that's present in the mice and the ALT values, or the amount of liver damage that's occurring. So on the y-axis here is the amount of MRP4 expression, and on the x-axis, the ALT that's present in the plasma, meaning that this is indicative of how much liver injury has occurred. So basically what this is demonstrating is that if there's a higher abundance of MRP4 in the tissue, there's a really low amount of liver injury that can be caused by acetaminophen. However, if MRP4 expression is really low and they're exposed to acetaminophen, the ALT values, or the amount of liver injury that occurs as a result of that, would be high. So there was a direct negative correlation between the amount of MRP4 and the amount of liver damage that can occur. So the interesting thing is that although we know there is this protective mechanism and that it does involve MRP4, we really still don't know how MRP4 is being protective against acetaminophen toxicity. And one of the reasons why we really don't know how it's being protective is because MRP4, if I haven't stated it already, stands for multi-drug resistance protein. So it's called a multi-drug resistance protein because it transports multiple different drugs. It has a wide variety of substrates, substrates being the chemicals that it transports or effluxes across that membrane. So this is a list of some of the substrates that MRP4 transports. So basically, this protein could be being upregulated in order to transport any one of these things that could be resulting in the protection. But what we don't know is which of these things it's being upregulated in order to expel from the hepatocyte during acetaminophen toxicity. Or maybe it's none of these things, and maybe it's a substrate that hasn't been identified yet for MRP4. So that's also a possibility. These are the currently known substrates, but that doesn't mean that they're the only substrates for this particular transporter. So unfortunately, we have kind of an incomplete story, although an interesting one. We have, a protein that is definitely important for providing some sort of protective mechanism, but because it has such a wide variety of substrates, it's really difficult to pinpoint exactly what that protein is doing or what it's effluxing that is serving as the protective mechanism. So that's kind of one part of the story of our lab. Another interesting aspect of the work that's been going on is with regard to how MRP4 expression is regulated. So I've talked about how the amount of MRP4 in the cell is increased when you give acetaminophen. But what we haven't talked about is how that increase actually occurs and what sort of regulatory pathways are in place that are actually causing that upregulation. So this schematic is showing essentially a hepatocyte. And when it's exposed to, there's a couple of effects that acetaminophen has. And what they've determined from previous work in our lab is that a transcription factor called NRF2 and also Kupffer cell function are really important for as pathways that control MRP4 expression in response to acetaminophen. So when these cells are exposed to acetaminophen, the Kupffer cell I haven't really mentioned yet, but it is essentially the immune cell of the liver, is one way to think of it. So when it's exposed to acetaminophen, it releases what are called cytokines. And cytokines can bind to receptors on the hepatocytes and produce a signaling pathway that would ultimately lead to an increase in transcription of MRP4 and an increase in the amount of protein that's present in the hepatocyte, or that's made in the cell. In addition to that, acetaminophen can also cause what's called oxidative stress in the hepatocyte. And this, we've shown, will induce a transcription factor called NRF2 and cause NRF2 to translocate to the nucleus of the cell, where it will activate transcription of a lot of different hepatoprotective genes. So MRP4 being one of them, but we've demonstrated that it also causes gene induction of many different other potentially protective genes. So this is kind of another avenue that has been pursued in our laboratory. And basically what it has, kind of the message that it has conveyed to us is that this adaptation that occurs in these cells is much more complex than, than you might hope, I guess. You would hope it would be really a simple story and easy to figure out. But the reality is that the adaptive responses here are multifactorial, meaning that there are probably, likely, multiple genes involved. So it's not just MRP4. We talked about that there were several uptake transporters which were downregulated, and there were many efflux transporters which were all upregulated. So it's not just one gene that's providing this protection, but rather a lot of different genes all kind of working together to provide some sort of protective mechanism. So one of the ways, so you might be thinking, all right, well, how do you figure out what genes, of all of the genes in the genome, how do you figure out which ones are, could be playing a role here and which ones aren't? So one of the ways that we approach something like that is doing what's called a transcriptomic study, or a gene array analysis. And basically this approach allows you to examine a wide range of genes and look for differential expression between groups. So in other words, I have this group of mice that's untreated, control mice, not treated with acetaminophen. Then I have another group of mice that have been treated with acetaminophen. And what I would like to do is compare the genes in those two mice, compare a wide variety of genes in those two mice, and determine what other genes are altered or changed because of that acetaminophen exposure. So that's essentially what a transcriptomic study does. It's a broad analysis of the RNA sequences in two different groups. I wrote of cells here, but liver tissue extrapolated to cells essentially. And it's basically used by researchers commonly to identify other genes of interest. So we know that this phenomena incorporates many different genes, but we don't know what they are. So in order to pursue some of those genes, we kind of need to get an idea of what genes could potentially be involved in this process. So one of Dr. Manichew's previous students performed one of these transcriptomic studies, and in the acetaminophen autoprotected mice, and did a gene array analysis. So what they have done here, so the dosing regimen is similar to what I just described to you on the previous slide, where the pre-treatment dose of the mice given at time 0 was either 400 mg per kg or vehicle, and then 48 hours later, those mice were either re-exposed to acetaminophen at 600 mg per kg, or they were given a vehicle. So this schematic provided 4 different treatment groups. So the first treatment group being mice that received vehicle at both doses, and then the second, so those would be termed VV. The second treatment group being mice that received first that initial acetaminophen group dose, and then received the vehicle 48 hours later. So this is an extra control group that was implied. The next group being a vehicle acetaminophen group, which received the pretreatment dose of vehicle and then the toxic dose of the acetaminophen. And then the final group being that autoprotected group, so the group of mice that received both doses of acetaminophen and exhibited that protection against them. So after this second exposure, there were mice that were sacrificed 4 hours after that second dose, and then there was a group of mice that was sacrificed 24 hours after that second dose. And what they did after sacrifice was harvest the liver and isolate RNA from the liver so that they could perform this gene array analysis. So I don't actually know how I'm doing on time. Do you? Is it 45 or so?
You can do about 5 minutes.
Okay. So basically, what the results of this gene array analysis show is that there's a lot of genes differentially expressed between these groups. So I'm kind of going to skip through some of what's shown here, and just kind of give you the punchline here. Basically, what they did was they eliminated all of the genes that were differentially expressed in the control groups, right? Because those are not relevant to the APAP autoprotection story. And what they resulted with was in the APAP autoprotected or AA group at 4 hours and the AA group at 24 hours, between these two groups, there were 13 genes in common that both of those groups had that were differentially expressed between those groups and the vehicle and other control groups. So these 13 genes were identified as genes of interest for being potentially related to that acetaminophen protection. So this is the ultimate result of this transcriptomic study, is that you get this list of genes, as you can see. So here are the gene symbols or the gene names. And their fold change, or the amount that expression changed of that gene between the vehicle controls and the APAP autoprotected group. So you get this really comprehensive list of genes that you can then go through and try and think about why those genes might be differentially expressed in that tissue, or what kind of mechanism those genes might be doing in order to provide that kind of protection. So this kind of gives maybe a little bit of like a jumping off point for researchers in terms of it gives you some clues as to what kind of avenues you want to pursue next. So for example, one of the graduate students currently in our lab has been pursuing this gene FMO3 as one of the potentially important genes for protecting against acetaminophen toxicity. And it's turned out to be a really interesting story. I won't go into it, but it turns out that this gene is not one that you would have commonly thought up based on just what we know about acetaminophen in the liver and toxicity, but it was identified in this gene array study. So without having this, we would have never been led to something like FMO3. So these are really useful tools that we employ now and will in the future for kind of helping us to progress our research forward. So I guess with that, I will obviously acknowledge first and foremost Dr. Manitou, whose work I've presented here today, the members of his lab, past and present, Dr. Alex Sunis, Dr. Campion, and Megan O'Connor were the main contributors to a lot of the work that was presented here today. And then these are more recent and current members of the lab who are kind of pursuing some of the work that followed this with regard to some of those other genes. My project is revolving around pursuing things regarding MRP4, past what I've talked about here. And we are supported by the National Institute of Health and the UConn School of Pharmacy. This right here, if you've never met him, is Dr. Manitou himself and some of our lab members. And these are actually two of Dr. Morris's graduate students as well that I've included in the picture. So, what's that?
They want free food.
They want free food, yeah. So I guess at this time I'll take any questions if you have them.
You've been talking a lot about acetaminophen. I'm curious if the toxicity is particular to acetaminophen, or are there other pain-relieving drugs like NSAIDs, like ibuprofen or naproxen sodium, that have similar profiles, or is this really acetaminophen specific?
So there are other drugs that have similar profiles, produce a similar toxicity as acetaminophen, carbon tetrachloride, and other compounds like that, but I don't think that the NSAIDs produce this type of toxicity. So what you're referring to is the non-steroidal anti-inflammatory drugs. The toxicity profile for those is different. As I've said, they are an anti-inflammatory drug rather than a just basically an analgesic like acetaminophen. So they do have different toxic profiles. Yeah.
Would this resistance be akin to development of tolerance or sensitivity to acetaminophen?
Yeah, it's definitely the case that they are more tolerant to the dose, to that second subsequent higher dose. So ordinarily, that second dose would be if when you were giving it to a mouse that wasn't pre-treated, it was a potentially lethal or very toxic dose of acetaminophen. But when they are pre-treated with it, they do develop a tolerance to it, and this we refer to it as protection against it. But yeah, essentially tolerance would be an accurate word as well. They just become more resistant to it for whatever reason.
How would the pre-treatment, or is there research on like what would that pre-treatment look like if you were to take it to human clinical trials?
In terms of the actual dose that's given here, this is a very high dose of acetaminophen given to these mice. It would be it's not something that they would ever do in a clinical trial because they wouldn't administer this high of a dose, an equivalent dose like this to a patient. As Dr. Morris was saying, if we use animal studies and we give a much higher dose a lot of the time than you would ever see in a human clinical trial. So in terms of an equivalent dose, I don't know the answer because I don't think that you do it. But I mean, there are people not necessarily involved in clinical trials, but who are taking super therapeutic doses of acetaminophen regularly for chronic pain or for whatever reason that do develop a resistance similar to this, and that has been seen in clinics. But it's not something where they can do a specific trial in a clinic that would be similar to this.
So then would the focus then be just sort of this gene study to take, is that, I guess, sort of past the mice stage as this is really, fascinating research, where does that lead to as far as developing something to look at sort of the liver toxicity that they're seeing in this?
Yeah, so the idea with doing, these animal studies and this transcriptomic study is that you could potentially get at the molecular mechanisms of the toxicity, right? So if we can learn more about how exactly the cell is responding, a lot of that can be extrapolated to human health and the actual toxicity that's occurring in the cells. And what genes are upregulated and stuff like that would be extrapolated to humans, theoretically. So that's kind of the idea, is to learn more about the molecular components of the protection so that we can, either develop maybe a drug that can be used in conjunction with acetaminophen that would, prevent the toxicity based on what we know about the molecular cause of the toxicity, or, something like that. Does that answer your question?
Yeah, no.
Okay. I don't know who first. It doesn't matter.
I'll ask a question. One thing that's of concern with acetaminophen is that it's often used to reduce fever in infants and young children.
Yeah.
And it's very tempting for parents to see that the fever is not coming down fast enough and to want to give more.
Yes.
And it's very worrisome in young children who are smaller and more immature livers. So I'm wondering, do you do any experimentation on mice of various ages and see differences in how they respond?
So most of our work is done in young adult mice. So we don't do any juvenile mice for our studies, but there is evidence and there is a lot known about the difference that can be occur in a child versus an adult in the way that they process acetaminophen and the way that the ability that they have to detoxify the chemical. So yeah, it is like a huge concern regarding giving children drugs like this, because they don't have the developed liver that adults have, and they don't have the same detoxification pathways that an adult liver would have. So yeah, it's definitely a concern, and it is tempting to not follow that dose that's right on the bottle, right? It says, all right, it says I should take 2 every 4 hours, but my headache's really bad, so I'm going to take 3, right? So a lot of people do that. And it's very tempting to do that, but ultimately that is the cause, the underlying cause of all of those unintentional overdoses that occur. So, yeah.
I was wondering how well understood the mechanism is for why it works as an analgesic, and whether that is affected by pre-treatment. You know, I mean, do people get used to a certain level of pain relief?
So do they get like tolerant of the therapy as well as the toxicity? I guess I don't. I guess my answer is I don't know the answer to that. With other drugs, you could say that that is absolutely true, and that people develop, an addiction and a tolerance to certain things, and they can, over time, after taking that drug, need more and more of it in order to feel the therapeutic effects of that. I don't know. Do you know, Dr. Morris, if that's true? Yeah, I'm not sure.
Mice squeak less loud.
I don't notice anything in the mice, but we also don't. Our studies aren't such that we give the mice, repeated doses over a long period of time where you would see such an effect. So I guess my answer is I'm not really sure. Yeah, either one of you.
So actually, I have two questions. So with respect to your upregulated genes from pre-treatment, I noticed you have hemoglobin genes that are showing up in both cases. Is that potentially an indication that what's being effluxed from hepatocytes is the toxic product of P450, and that's then acting on red blood cells in the bloodstream, so that the protective effect is not something that is being kicked out of the cell to intercept the acetaminophen in the bloodstream, but actually it's toxic byproduct to give the liver time to replenish its stores so that it can process.
So that's an interesting hypothesis, and I don't know if it's true or not, or it's not something that our lab has pursued. With regard, you're talking about these two genes, I assume here.
I was wondering if that was something so ridiculous that you could discount it out of hand, or if that's actually.
Off the top of my head, I can't completely discount it, but I can say that it's not something that I know enough about to discount or say that it would be true. But we are examining this list of genes that were resulted from our transcriptomic studies, and as we get to some of the genes, maybe something interesting along those lines would pop up. So that's kind of the really cool thing about these transcriptomic studies, is that they present genes that you may never even have thought of that had a possible potential to be a part of this protection pathway. So.
The other question was, so you would characterize this as a type A
Yeah,
reaction?
so acetaminophen would be a type A
So
reaction.
To touch on the type B reactions for a second and tie that back into our discussion last night, which was about personalized medicine. So those sorts of low frequency incidences at therapeutic doses, are those the sorts of things that you would recommend when those happen in the clinical population? Is that the sort of thing that personalized medicine could address, maybe by sequencing the major histocompatibility complex and looking for common markers that might indicate a predilection to an immune reaction or an allergic reaction to?
Yeah, definitely. That is, ultimately the goal of personalized medicine, is that it is personalized and it's specific for individuals. And those type B reactions are reactions that are not predictable. They occur in a very small subset of the population that is, just for whatever reason, genetically different and more susceptible to that drug. So, the idea of personalized medicine would, in analyzing, each individual's genes before giving them a toxic chemical, to try and predict, using modeling systems and such, how their body would react, perhaps adversely, to a drug, is a really useful tool. And that's really cool that you guys learned about that last night. So, yeah.
I'm curious, predosing the animals with acetaminophen prevents hepatotoxicity. Does it do anything for the kidneys? Because you talked about corollary kidney damage.
Yeah, so the kid.
Is it protecting the kidney as well, or are you getting kidney damage at the high doses?
So so I did mention that the kidney is the secondary organ of toxicity with acetaminophen. I don't know of any animal studies that have shown this autoprotective mechanism, but I have done some work in isolated proximal tubule cells from the kidney and acetaminophen, and those indicate that there is potentially a protective mechanism there as well, meaning that when they get this pretreatment dose. But you have to keep in mind that a lot of the same transporters that I talked about, I called them hepatobiliary transporters, but a lot of them are also present in kidney tissue and are going to be effluxing things in the proximal tubule and the distal tubule of the kidney. So there is definitely potential for an overlapping of the mechanism between the two organ systems. So they're both excretory organs. The kidney doesn't have the metabolic capabilities that the liver has in terms of detoxification, but does have a lot of those same transporters and is really important for excretion. So there is certainly the possibility that there is some level of protection in the kidney as well, just based on the idea that some of those transporters could be altered in an acetaminophen. Yeah, it's not really clear at this point, but it's definitely something that is pursued.
Just a basic question. Does the body, do these animals ever excrete non-conjugated acetaminophen, or is it always?
Is it all detoxified? No, some of it is excreted. A small percentage of it is excreted as the parent compound. And then the majority of it is excreted as those glucuronide and sulfated conjugates that I talked about. But yeah, there is some of the parent compound that comes that gets eliminated as well. But the capacity to eliminate the parent compound is relatively low.
Thank you. So, one of the things that came out, as Carolyn mentioned earlier, is that there was a desire to have a little more information from our speakers about how they currently look for evidence and position and support their work, manage their research data, and share research data findings. So, I just going to kick it off with the same question Carolyn asked earlier, which is, how effective are your faculty and student literature search skills, and what needs improvement? And then we'll let everybody else ask whatever seems relevant.
Well, we wouldn't know if they're not effective because we don't know what we didn't find.
Go ahead.
It sort of depends on the questions we're asking you. Where do I go? I go to PubMed. I can find most anything I want in PubMed.
Oh, yeah, sorry.
Can I take one and you take the other, or how do we do it? Pull this way.
Yeah, I start off in PubMed, and I can usually find what I want in PubMed. There are tox-specific searches that you can do, specific toxic chemical searches, and you can do that. There's some information that's very hard to find, and that would be an obscure government document. It'd be pretty hard to find. I usually phone them and say, where is it? Or I share them. The other thing in the tox world is there's a huge repository of unpublished data, and as a profession, actually, we're trying to deal with what do we do about unpublished data. For every compound that made it to the market as a drug, there are 10,000 that failed because of toxicity, and the drug companies have huge databases. And they're all overlapping, but they don't share them because they're proprietary, and they would just assume the other drug company goes down this dead end. And so the real issue, seriously, as a professional, how can we make big databases available for unpublished information? So that's really one of the challenges right now. In the old days, I used to go to Chem Abstract, and do those 5-year catalogs and search it that way, but now it's just a long line. And rarely am I caught by something I haven't seen.
Yeah, I don't know if I have much to add, but I also typically first go to PubMed. I do all my searching electronically. I don't really use any hard copy journals anymore. Occasionally I can't get something on PubMed, and that's why we have interlibrary loan, which is wonderful, but for the most part I find pretty much everything I need. And if I'm not finding everything I need, I don't know it because I'm not finding it. So,
Yeah.
If you could go back to your just beginning grad school self and say, I should have known this way when I started, my life would have been so much easier, what would it be?
What's
Find
it-
an online search. In 1973. We have a wonderful librarian who actually teaches search skills to all our students. So
I don't know if I can really answer that because my graduate days were only a few years ago, and all of the same tools that are that I would use now I used back then also. So I don't know that I have much insight in terms of that. But, yeah.
Well, I tell my kids not to use Google.
Yeah.
Not when I'm looking. So a more reputable source. So Google pulls up most everything you'll find in PubMed, I must agree.
Yeah, the younger generation, I see it with the new graduate students coming into our lab, are using a lot of Google Scholar. So I haven't, just because I'm so familiar with PubMed. I'm kind of stuck in what's familiar, but I do notice that there is a large number of students now using Google Scholar instead of PubMed. So.
How do you track your citations? How do you store them? How do you organize them?
I use RefWorks because it's free through the university, but Dr. Morris is a little more distinguished and maybe uses something different. But
yeah, I know it's all electronic.
Yeah, you use RefWorks or?
EndNote or
EndNote, yeah.
they're all interchangeable now. You can move from one platform to the other really easily and move all of your citations. So like differences. EndNote isn't free, so.
So, the real hang-up is not storing it in my computer, but it's that every journal has a different style for putting out the references. And so the real nice thing is having the EndNote just switch the format.
Yeah.
That, to me, that's the biggest convenience. And actually, most of the historic references that I need for my line of work, I already have. They're all stored in my computer, so I don't need to pick up the news. They're already there. And I'm actually so archaic in my computer, I actually have it all in a huge Excel spreadsheet with 3 keywords for each of the author, and I can find anything in literally 30 seconds. If it's in my Excel spreadsheet, I can find it. Yeah.
You mentioned this disconnect between what's happening in the scientific community, what's happening in journalism, and then what's happening in policy. Do you see any way to remedy that, or do you have any ideas? Even pie in the sky, I'll take that too.
Only elect PhDs in Congress. I don't that would be worse. There's a level of distrust of highly educated people, and I think we need to work on building up trust and relying on the judgment of an expert in the field, no matter what that field is. And I think that level of distrust didn't used to exist 40 years ago, and if we could work on that, I think from both sides, I think the scientists need to be a little more even-handed, perhaps. And I think that's actually going to be the key. No one knows everything, so we've got to trust some people. Right, way in the back.
Hi, are you familiar with ToxNet or ToxLit? And if you are, do you find them easy to use or cumbersome, or PubMed has everything?
I'm familiar with using them. I used to. I don't do it anymore because I just use PubMed. So I'm really not up to date on those, to be honest. They might be lovely, and I'm just a creature of habit.
How are pharmacy journals on submitting some of your data? So a lot of journals in college and other areas now require that you
We're evolving now, actually journals are going to want the original data, the primary data. And most of the toxicology and the pharmacy journals aren't there yet and requiring them. But you know, NIH is, and I think actually that's one of my biggest concerns is how are we going to make a repository of all of my original data? That's what I'm asking you guys. Right? Well, at some place where my original data can be stored where someone can find it and retrieve it. Exactly. Exactly. And that's at the university. I know this is what we're actually starting to talk about with the library is how can they help us do that. Yeah.
Do you use electronic lab notebooks? Is there a reason?
I tried once for a period in graduate school. I tried to use an electronic notebook, and I found it to be really difficult. Mostly because you know, a lot of times I'm at my bench and write something. I mean, I found myself keeping an actual notebook and then also an electronic notebook, and it didn't seem efficient to me. But I'm sure other people have better methods of doing it. I don't know. It definitely works for some people.
That's what in my lab. There's a hard copy lab book that's on the bench. Everything is there. That's the Bible. Data is taken from there and then put into a computer and then stored electronically as well. But I can tell you, there have been times when I've gone back, 15 years, and I had to find the book for a little note about, oh yeah, this animal or whatever. And that way I'm old-fashioned. I suppose we could have an iPad right next to us and do it that way. But you know, I'm just old-fashioned. You do the book. You got the book forever.
Yeah, iPad can break.
Yeah, you know, I mean, even like CDs are out of date now. Right? Flash drives will be out of date. I'm just going to want to change the format.
Yeah.
So I'm horribly old-fashioned. It's in a notebook and a pen with no erasures.
What skills do you feel, do you see lacking in students, whether they're undergraduate or graduate school research, that you see are lacking or that you need, that perhaps the library can help develop in collaboration?
That was okay until you got to the last part. I'm older, so I can talk about the older generation and how much better we were back in the day. So there's a guy on my hockey team, and he has a shirt that says, "The older I am, the better I was." That's my life.
Or what skills in general, in research?
I mean, the answer, before you got to the library part, but maybe you can put this back to me. Quantitative mathematical skills. Mathematically based logical skills, I don't think are anywhere near as strong now as they used to be. How libraries can help with that, I'm open to any sort of suggestion.
Well, I don't know, maybe Carolyn, you can speak to this. We don't have anything on that.
Okay.
We, you know, at least at our library, we have a librarian who helps with like R, teaching R, teaching Vivo, all of these different types of qualitative, quantitative stats programs. So, it's the beginning.
Yeah, we could spend a long time talking about this. It's sort of be in my bonnet. And, you know, most of what I read in science journals say actually the problem is back in elementary school and middle school. Fundamental conceptual mathematics skills aren't ingrained at that point, and they're actually saying it's too late. Yeah.
I'm wondering if you have any thoughts or comments about research that's done in the private sector as opposed to the academic world.
Yes.
That you could share.
That I could share. You know, there are excellent scientists in the private sector. I don't have a, you know, they're as good as scientists in academia. I totally believe all their data. Right? You look at the data. I mean, I can look at the data. I might form a slightly different conclusion from the data, but the primary data is totally sound. This is with any scientist. I think we're going to be moving forward. We're creating more PhDs then they will find jobs in academia and more of them go to the private sector. There's nothing, I have nothing wrong with the private sector whatsoever. I trust them just as much. So I don't have a problem with that. With conflict of interest or bias, I don't share those concerns. They're, if they have a bias, it's readily clear.
It's what?
Readily clear, right? We all have biases. I have my little pet research area, and you got a problem in that area, I know the lab that can do it. And boy, you really need this data. People in academia have biases as well. So that doesn't bother me. In the toxicology world, there's a lot of wonderful science going on in the private sector. And they have access to huge amounts of data that we don't, because it's proprietary. Yeah.
So I've heard that a lot of the data that's out there, the negative data, is not, you just mentioned, I've heard in other places that it's not published, and that actually would be very useful for researchers, so people are not repeating the same things over and over again. Is there any, are you aware of any interest or attempts that might possibly succeed at some point in making that available? How could people in this area go about getting that information and making it available?
I'm aware of attempts. Whether it's going to be successful or not. I have my Society of Toxicology hat on. We actually got a task force that's actually looking at exactly that. How do we make these data available? How do we get together a common repository for this sort of information? No one's got easy answers to those questions because a lot of it are trade secrets and proprietary, and this company paid to generate this data and doesn't necessarily want to share it. But there are, at an organizational level, there are attempts to try to overcome these barriers. That's where we are right now, is just trying to come up with the attempts. In the toxicological world, this is really an inherent problem because from a safety assessment viewpoint, you'd like to determine the dose that doesn't do anything in an animal. You can't publish that. And so a priori, those sorts of data don't really get published. And that's a huge problem. One we're trying to address, but there are no simple answers. Yep.
If I'm correct, the research says that about 30 to 50% of all liver toxicity cases are due to acetaminophen. But then you mentioned that it seems that the policy is towards educating people not to use too much Tylenol or so forth. But there's no changes to the prescription drugs, Percocet and so forth. Why would the policy lean that way? Is it money? Is it government? Is it science? Why not put any regulations on those prescription drugs, which are heavily abused?
Well, there's a policy basis for that, and that is that the FDA assesses the risk and the benefit at the prescribed doses. It does not specifically, set tolerances based on overdoses, because then we wouldn't have a single drug on the market in the country. And so they are aware of the concern, but they're not basing the approval or the disapproval of the drug on what might happen if you abuse it. So that's why that's occurring. But they are making changes, lowering the acetaminophen in Percocet, right, and some of the opioid drugs, and they're probably moving. I want to make a prediction: in 5 years they'll get all of the acetaminophen out of there. Their drug companies are now actually putting forward forms of those drugs with no acetaminophen in it. Okay, specifically, so they are dealing with that. They're getting the bandwagon. Nothing is safe. Right? So even at over-the-counter drugs, there are risks associated with acetaminophen. These are those risks.